Thank you, Rob. What a lovely introduction. I had a moment of panic. I thought you were going to have pictures from when I was playing back in the 80s. Thank God you didn't. Let's see. There it is. Oops, no, that's the town hall. Oh, you already put it up for me? Fantastic. Okay, and as Rob said, actually, I love the last talk, and I thought, I'm smiling because I'm not a neuroscientist. I'm smiling because I'm a human geneticist, and actually it's turned out that identifying genes, while a difficult problem, has been a tractable problem of late, and turns out, I think, to be the easiest part in thinking about getting from genes to an understanding of complex human behavior. So I'm going to talk to you about that, about both some of the success the field has really transformed over the last 15 years or so, and then also talk about sort of some of the challenges of taking the next step to really go from an understanding of genes, not just to an understanding of biology, but truly to an understanding of the underlying pathophysiology in a way that hopefully will lead to being able to help our patients and their families. So I'm going to talk a little bit about autism and then Tourette syndrome, and then bring it together to address some of the issues of the challenges going forward. So autism is a neurodevelopmental syndrome that's characterized by fundamental deficits in reciprocal social communication, and as well as repetitive behaviors and highly restricted interests. The diagnostic formulation has changed slightly with successive courses of the DSM, but really the sine qua non is this impairment in the ability to interpret and to extend social communication. We've known for decades that there's a marked contribution from genetics, from family and twin studies, but really prior to the turn of the millennium, the search for genes that were contributing to common forms of the disorder, things that we would see every day in clinic, really was almost, it was completely stymied. And it was, as Rob pointed out, I was a clinical child psychiatrist and made a decision to go back to get a PhD in genetics because of, in part, the frustration with being in the clinics with families and with kids who were suffering some severe neurodevelopmental disorders, and really feeling as though our complete absence at that time, really in the late 1990s, of any understanding of the molecular cellular circuit level pathobiology was profoundly limiting, not only in our ability to tell families what was going wrong, but to think in any sort of creative ways about how to do better with treatments. And the thought that I had, which I have now pursued for 20 years, was that gene discovery would be one avenue to do this, with the idea that if you could reliably identify genes that were leading to the disorders that we were interested in, that would get you immediately to the molecular level, and then hopefully you could claw your way back to understanding something important about the pathophysiology. So as I said, prior to the turn of the millennium, there was not a lot of success, but there were profound changes at around that time. First, the sequencing of the human genome, and then importantly, the development of tools that allowed us to look with much higher resolution at DNA, which really empowered genetic studies in a profoundly important way. So there were a couple of important observations that I want to convey that I think really crystallized and was a harbinger for all of the success that's happened over the last decade. So one was that even in the late 90s, it was clear that if you looked at kids who had autism, that there were a greater number of gross chromosomal abnormalities compared to kids without autism, and that included new or de novo chromosomal abnormalities. And about 2007, there was a profoundly important paper from Jonathan Sabot and Mike Wigler at Cold Spring Harbor, in which they used new microarray technology, brand new, hot off the shelf, in order to look not at gross chromosomal abnormalities, but at submicroscopic chromosomal abnormalities, which they called copy number variations. And they sifted through the DNA of kids with autism compared to controls with a particular focus. And the focus was on new mutation, on de novo mutation. And what they found was that there was an increased rate of de novo submicroscopic copy number variations in kids with autism versus compared to kids without. That became the inspiration for something called the Simon Simplex collection. Jim Simon's a wealthy philanthropist in New York, saw this as an opportunity and partnered with Mike Wigler and others to begin to think about how you could execute on developing a cohort of families that would be particularly amenable to finding new mutation. And what they ended up with was a design like this. So they set out to collect 3 about 3,000 families in which parents would be unaffected. There would only be a single affected child, and if there were siblings, they would be unaffected. And the idea here is that this wouldn't guarantee that it was a new mutation, but it would enrich for finding new mutations in these families. And about 80% of the Simon Simplex collection actually had siblings. About 20% was just a parent-child trio. A couple other things I want to point out before getting into the data is that for de novo mutation arises, if it is going to turn out to be germline de novo mutation, in sperm or egg right before fertilization. And because of that, the mutation is not present in the parents, but is present in every cell in the child. Now, one of the interesting things about that is that it gives natural selection almost no time to weed that mutation out of the population, right? So if the child comes to term and is born, then even if there are very large effects on brain development, social functioning, things that otherwise you might expect would decrease fitness over multiple generations, you'll be able to identify those because there's no time for natural selection to act on them. So one of the things that we were most interested in, inspired by the strategy that Rick Lifton had taken in other disorders, was to look for very high effect mutations, with the idea that if you wanted to use genetics as an entree into biology, the bigger the biological effect, the more likely it would be that you could take that to the bench and find out something important about pathophysiology. So de novo mutations were kind of theoretically a great place to look, and then Wigler and Sabat showed us that actually it might be quite profitable to be digging around there. And so I was very fortunate then to be asked to lead a consortium of geneticists to get the first look at the Simon Simplex collection, initially on about 1,000 families and then about 3,000 families. And the first set of studies that we did were really just to repeat the experiment that Wigler and Sabat did on just total about a couple hundred families, and look for submicroscopic copy number variations and ask the question, do you see greater numbers in kids from what are called simplex families, simplex meaning only one a child with autism in the family, versus actually their siblings, which we used as a wonderful control for genetic study. And what we found, this just shows you that kind of regardless of how you count, and whether you're looking at the first group of about 1,000 families or the second group of about 2,500 families, is there's a far greater rate of copy number variations, both duplications and deletions in kids with autism versus those without. Now, these are very rare events. Importantly, de novo mutation happens in every generation, but at a very low base rate. So you see less than one of these copy number variations that we're measuring here per generation. So these are individual events, but there are many more of them happening in kids with ASD versus kids without. And what we were able to kind of immediately discern from this is that not only is there a group difference, but if you then study the kids and begin to ask the question, is there any spot in the genome where these changes, either losses and gains in chromosomes, started lining up in the genome, that would be a marker that you were looking at a risk event. We worked hard to develop kind of rigorous statistical frameworks to make sure that what we were seeing was not just by chance. Lightning does strike twice by chance every once in a while. So we had some wonderful statistical collaborators, Bernie Devil and Catherine Rader, and we developed an approach to be able to identify regions of the genome in which these copy number variations were present, as I said, more likely than would be expected by chance, and to correct for all of the comparisons that we would see across the genome. This is a table published. Stephen Sanders was, at the time, a postdoc in the lab. Now he's running his own lab at UCSF, listing sort of the cumulative data, not only from the 3,000 families or so in the Simon Simplex collection, but by the time we published this table, about 2,000 other families that have been studied by other large groups. And we can now begin to give you a list of chromosomal regions, relatively small losses and gains in chromosomal regions that are statistically associated with ASD. What I want to do just is to digress for a minute just to talk overall about this region. We found that duplications of 7q11.23, that's just a marker of a location on a chromosome, were associated with classic autism. This is a child who shows that phenotype. I was talking through it, but perhaps if you were looking at the slide, you saw repetitive behaviors with hand flapping. You don't have to worry about the sound. There really isn't any anything that you need to hear here. But one of the reasons actually I use it, even though there isn't sound, is that I don't think it takes any clinical training at all to sense that the social relatedness of that child to the two other people in the room is unusual. And then when you know that that's his father and that's an examiner, this is not a typically developing 13-year-old boy in terms of, you can imagine the way that a typically developing person might interact in the room. And that kind of fundamental impairment, I think, that's quite obvious here is, as I said, the hallmark of ASD. But I want to move on, and what I want to show you now is the phenotype that's associated not with gains, what we identified here, but it had already been determined that losses of this exact same chromosomal region, exactly the same breakpoints, exactly the same genes, losses instead of gains were already known to lead to a neurodevelopmental disorder. And I'm going to show you that here. Here we go. Let's see if this works.
Here wants to be your friend.
Yes, nice to meet you, bud.
Nice to meet you. You're great. You're great. You got great hands.
Hi, Chris.
Hello, buddy. How are you?
I'm really watching on ABC News.
Oh, thank you. Thank you.
What's your favorite color and what's your favorite TV show?
Kids love to ask questions, but not as much as these kids.
My favorite color is blue. I have met Barney the dinosaur.
Yeah. Where do you live?
I live in New York City.
Do you have any sons or daughters?
I do.
Wow.
I have 2 daughters and a son.
And kids love to make friends, but not like these kids.
What nationality are you?
I'm Italian. Oh.
How do you, buddy?
They look a little different, but what makes them special is a rare condition called Williams syndrome.
So
Can you be
Williams syndrome.
born-
Whoops, stop. 2020, second only to Wikipedia, is a great source of scientific information. Williams syndrome, exactly the same chromosomal region. We found duplications led to autism. This is if you looked at that, I hope again that you wouldn't need any clinical training at all to sense this intense interest in reciprocal social communication among the kids, that every single one of the kids that you just saw, it is a highly reliable behavioral phenotype. It is in many respects kind of how people who clinically see Williams kids think about them, is that it's this kind of hyper-social behavioral phenotype. There are a variety of other very interesting behavioral aspects of Williams syndromes, but one of the reasons that I raise it is that for us in the laboratory, it was really a great motivator. When you think about our hypothesis, it was we can sort through the DNA, identify a change at the level of DNA that is going to have predictable and reliable enough relationship to complex human behavior after, the DNA has to turn into a brain, the brain has to develop, it has to live in society, it has all kinds of environmental inputs, and the notion that you could find single genetic lesions that would give you enough traction to understand something as complex as social behavior. This is just kind of an experiment in nature, but I think it begins to suggest, if you do the thought experiment, if you knew what was in that region of 25 genes that so profoundly altered people's interest in social affiliation, that potentially could give you a really important answer about pathophysiology of autism spectrum disorders. Now, so we love that finding. Again, as I said, it gave us a lot of energy, but there were problems. So the purpose of our lab was to find genes to be certain that they were contributing to the disorder we're interested in, and then pass them off to folks like Rob and other collaborators to try to understand what they were doing. And when you go to someone like Rob and say, there are 25 genes in this interval, and unfortunately, I can't tell you whether or not it's one gene. Actually, we have a lot of evidence that suggests it's not just one gene. We think it's multiple genes, and it may be different combinations within those 25. So can you model that and tell us about pathophysiology? Not a great experiment. Even with CRISPR, not a great set of experiments. What would be great would be to be able to do exactly the same thing with a single gene. And fortunately, around 2011, we got the next wave of genomic technology, which was the ability to assay essentially every base in the coding portion of the genome through exome sequencing. So that was developed kind of in parallel a number of places. I was fortunate enough to be next door to Rick Lifton, who was working hard on getting that up and running. And then we were able to turn it very quickly after development of the technology to look at the Simon Simplex collection again. Exactly the same experiment. And what we found is that just like copy number variations, we saw an increased rate of what are called likely gene-disrupting de novo mutations in kids with autism, ASD, versus their sibling, again, at great control. Now, these are de novo mutations, but you can see that they're of a particular type here. These are mutations that would be expected to alter a protein structure or function. And so we call them, as I said, likely gene-disrupting, or they're sometimes called putatively loss-of-function mutations. So the subset of mutations is overrepresented in autism spectrum disorders. What I want to show you is if you look, and now using, this is jumping a bit later, but using whole genome sequencing, if you ask the question, because the vast majority even of de novo mutations are not this small set of likely gene disrupting, if you just look across kind of all de novo mutations in cases versus the controls, you see no difference. In addition, if you ask the question, do kids with autism, are they more likely to have 2, 3, 4, 5 de novo mutations, likely gene disrupting, versus their sibling controls, you don't see any difference. This distribution is exactly the same between cases and controls. So what this is telling you is that this is not hypermutability, it's not randomly messing with the genome that's leading kids to have autism, and that it's not an accumulation of multiple kind of smaller risks. These are single large event or large effect, likely gene-disrupting mutations that are leading to this result of the overrepresentation in cases versus controls. And just like with copy number variation, we can develop a statistical framework that allows us to differentiate between seeing multiple of those mutations in the same place in the genome by chance. If we get enough of them lining up in the genome, then we can tell you exactly how unlikely that is to be by chance. And we can start to tell you with this kind of statistical framework either what the p-value or the false discovery rate is. We tend to like false discovery rate, but it would be a whole hour of lecture about why we think that's better than just a p-value. But in any case, for those of you who like kind of traditional kind of frequentist approaches, actually this group essentially is equivalent to a p-value that's corrected for genome-wide significance. Again, this is Stephen Sanders leading the way in the lab. And I just want to point out that, it sounds like just my lab doing this, but in fact, at every step of the way, we were kind of stride for stride with a series of other labs around the country, particularly Mark Daly in Boston, Mike Wigler at Cold Spring Harbor, and Evan Eichler in Washington. So this begins to give us kind of now a repository of genes. As I said, we'd call these the highest confidence genes, and these kind of probable autism genes because they have a greater than 90% chance, if you take an FDR of 0.1 and below, of being autism genes. And I'm going to switch gears a tiny bit here and talk a little bit about Tourette syndrome. So again, I'm showing you a video, and people often think about Tourette as just people, using profanity in an uncontrolled way. But in fact, the hallmarks are chronic vocal and motor tics, and you're seeing kind of the most common motor tics actually on display here, which is grunting or blowing air out through your nose, etc. Sometimes throat clearing. Only about 10% of people with Tourette syndrome who come to the clinic actually have coprolalia or use profanity in an uncontrolled way. You can also see that this is, a 20-year-old man, and both the vocal and motor tics are quite debilitating. While it is true that many people with Tourette syndrome get better, about 70% will improve across the lifespan, and particularly as they enter adulthood, it still is a disabling disorder for many, and it's highly, highly related to a couple of other neurodevelopmental disorders that cause a lot of difficulty in childhood, and quite commonly, like attention deficit hyperactivity disorder and OCD. So this story is going to be faster because I've already set it up with the autism work. We're about at least 5 years behind, really sadly only because it was hard to convince people to give us the money to repeat the experiments. But we just recently have been able to get to about 500 trios. And this is exactly the same experiment. We're just turning the crank. As I said, it's easier to be a geneticist sometimes than a neuroscientist. And you can see an excess in the rate of likely gene-disrupting de novo mutations in kids with Tourette versus without. We actually, and from this, it was just recently published in Neuron, and we can begin to see genes that are starting to kind of approach the high confidence realm here. These are p-values and FDR values. And then actually, we've now, in the last few months, been able to do about 300 more families, and now we clearly have 2 high confidence genes. And really, we can predict quite accurately now that if we can sequence more families, that will have the same ability ability to begin to accumulate both genes. These are copy number variations, so all this is showing is kind of in backward sequence exactly what we found in autism. So for both autism and Tourette syndrome now, there's a clear path forward to leverage de novo mutation. I want to just stop for a second and say this doesn't mean that de novo mutation accounts for the majority of genetic risk or is in some way more important than other types. There are lots of other ways that the genome can contribute to a common disorder. What it is a particularly tractable way to identify large effect mutations and ones that map to specific genes in the genome. So I want to talk about sort of where that leaves us. On the one hand, it is extraordinary. I've been doing it for 20 years or so, and to be able to stand here and to give you a list now of 71 autism loci, about 30 of them that we, are essentially definitively identified as ASD risk, another, 40 that are probable with sort of 90% or better probability. But that really is, in fact, just the first step of what we wanted to do in the lab, which was not only begin to identify these specific genetic markers, but then to be able to do something. And initially, this was my thought about what we were going to do, which was find a rare mutation carrying large effects and kind of do, this is some version of this is in every translational medicine talk ever, that you find the gene, then you study it, you move to model systems, you elaborate pathways, and that's going to take you to novel therapeutics. And boy, it would be great if it were that easy. I showed that for about 15 years and never really spent a lot of time thinking about it because we couldn't find any genes. And then when we found genes, I realized, oh my God, how embarrassing. So first of all, I'm a child psychiatrist, right? And so if you look at that, there are a couple things that are really glaringly missing from that. So one is brain and the other is development, right? It's this very static, would be good, and actually you can imagine where it came from, would be good maybe for a kidney, a developing pump. But when you think about the complexity of human brain and the biology, the notion that there would be any kind of one-to-one correspondence between a single pathway, that we could identify just by pulling on a single gene, and that would lead us to understanding something quite that complex, was sort of blissful ignorance. As soon as we began to discover genes, we started to think, kind of what are the sort of necessary steps that we need to follow now to begin to kind of set up the experiments for our neurobiological colleagues. What would be a way to do more than just simply saying, okay, here's a list of genes and you can go do each one one at a time? And the reason that doesn't feel that satisfying is that if you do that, because of the complexity of brain and because of the complexity of brain development, I think you can be guaranteed that you'd learn a boatload about biology, but I'm not sure that you would ever be able to know when you'd learned a boatload about pathophysiology from those kinds of experiments. If you start messing with genes that are involved in brain development or synaptic function or something like that, and you start looking all over the brain, you're going to be able to find a whole variety of phenotypes, some of which are potentially highly relevant to your disorder, some of which may be completely irrelevant to your disorder. So we started to think about sort of how to address that problem, and clearly kind of the simplest way to do it is to say, well, let's just take all of them together and see whether or not there's any signal that begins to tell us something about sort of triangulation or convergence. The notion, right, is that we may have many different mutations all leading in the same direction. Something is bringing those things together so that clinicians can reliably identify a set of complex behaviors in kids with autism. And so what falls out very easily without even any statistics is that it points to the synapse and it points to chromatin modification, transcriptional regulation, and the regulation of when genes are turned on and turned off. So now, on the one hand, there are people who look at this and say, my God, this is the most expensive penetrating glimpse into the obvious in the history of modern science. We've taken a neurodevelopmental disorder, spent tens of millions of dollars, and I'm showing you neuro and development, right? So is potentially problematic. The flip side, though, is I think that that's a little bit of an overly superficial read. The bottom line is it's not every molecule within the synapse. There are particular protein-protein interactions. There are particular synapses that are more likely to house a number of genes that are contributing to autism than other types of synapses. And we haven't gotten there yet, but there, chromatin modification is a highly regulated process. And so I think until we dig into what the genes are doing in these things, I think it may be a little facile to say, this is going to be a dead end. But it does, again, raise this problem of, when you start to pull on the thread of these genes, you're starting to run into something that looks like all of biology. And so then trying to figure out kind of how do you differentiate that from something that we're particularly interested in, which is, is there tractable knowledge here to develop new therapies? So we began to think in a slightly different way about this, and that really came out of a series of conversations with Nenad Sesten, my close collaborator at Yale, who at the time was just thinking about sort of normal human brain development and building one of the first maps of gene expression across time and across the various regions of developing human brain. So he was just really interested in, like, how does a normal brain get built with regard to gene expression? And obviously we had an interesting but quite orthogonal data set, which is a bunch of genes that we had a lot of confidence in were leading to autism. And so we thought, why don't we see whether or not we can combine those two things together? That instead of asking, what are these genes doing, maybe we can ask the question, when and where are they doing it? Is there some space in developing brain and time in developing brain in which these genes that we've identified that are highly disparate, they're in every cell compartment, they subserve a whole variety of different functions. Is there any temporal spatial convergence with regard to these genes? And we did it really, as I said, just by kind of the Reese's peanut butter cup approach. We took his chocolate and our peanut butter and we put it together. So his chocolate is a bunch of information on clusters of human brain region gene expression, and then this is over time in windows of time. And then we took our genes that we were most interested in autism and just looked to see what kind of the networks, the closest, most coherent networks of gene activity were around the autism genes that we were interested in. So what we built is for every brain region at every point in time. Just want to point out, this is birth. This is about 4 weeks of age. This is 80 years of age. So we got a lot of data prenatally, and then a lot less data postnatally. So far from a perfect data set. But we looked at all of these, so we had a gene expression, co-expression network for every brain region in every one of these time windows. And then we asked the question, if we take a separate set of autism genes that were not used to create these networks and ask the question, are they enriched for any of those networks? It would begin to suggest that we were pulling out something with regard to spatial temporal distribution that was highly relevant for autism. And in fact, we found out, starting initially, this was very early on, with only 9 high-confidence genes and about 130 probable genes, that the strongest signal that we got, this is prefrontal cortex predominantly, and this corresponds to about 18 to 24 weeks of gestation. So we're looking at mid-fetal prefrontal cortex. Once we were able to identify that network that was enriched for autism genes based on our statistics, we were then able to ask, what do those networks look like? Do they correspond to any particular cell type? At this point, we were, this is not single-cell data, we're simply using a preordained marker lists of genes that are expressed in each of these different cell types, and what we found, this is about 2013, was that the strongest enrichment was really for deep layer cortical glutamatergic neurons, so layer 5, layer 6, in developing midfetal PFC. As we moved on, and we have many more genes, actually, this is now a second set of 13 high-confidence genes, now increasing to 269 probable genes. And the kind of best signal shifts, but only slightly. It's still within the same kind of time window and remains in PFC and also remains in deep layers. And then actually the slide that I'm not going to show then takes all of the genes that I showed you on that table together and gets essentially the identical result. So what this is telling us is not a unifying theory of autism. Essentially all that it's saying is, for the genes that we've discovered through the process of using de novo mutation, which may be biologically a different kind of animal than other ways to get autism. But if you look at those genes in particular, they appear to be converging within a particular type of cells or set of cells. We're trying to narrow that down at a particular point in time, with the idea being that if you want to begin then to talk with your neurobiological colleagues about where you might look for convergent phenotype, whether that's electrophysiological, molecular, et cetera, we can at least give you some idea about where you might hypothesize that you would want to look for some convergence at that kind of functional level, which may map onto the convergence, the developmental convergence. And this is just for the non-neuroscientists, it's just a graphic representation of what we found. We took gene expression and de novo mutations, found an enriched network, and then we're able to map that network to developing brain and at particular layers of the cortex. And this is just meant to show that from a functional perspective, there's no convergence in these genes. They are in every cell. I mean, they are in every compartment of the cell, and they do very different things. But spatially and temporally, they are at least raising the possibility that the convergent phenotype in ASD has as much to do with when you perturb the development of circuits and which circuits you're perturbing as opposed to a specific molecular pathway. So I'm going to end just by saying that these de novo mutations in sequence and the structure of the DNA carry large risks for ASD and Tourette. I want to point out that this does not mean genes are fate. When I say large effects, the group that I showed you that's highest confidence for autism have collectively 20-fold increases in risk. Those orders of magnitude greater than you're used to reading in most of your genetics papers, where you might be looking at an allele that's increasing risk by 5 or 10%. These are 20, 30, 50-fold, sort of getting near what you would expect for Mendelian mutations, but they don't act in a Mendelian way. The way they are predisposing to neurodevelopmental outcomes, and not just a single one. We can have the same mutation that is clearly and reliably associated with ASD, and we turn to our colleagues in epilepsy, intellectual disability, schizophrenia, specific language impairment, and they're finding many of the same genes. It's not completely overlapping, but again, what you're beginning to see is sort of that genes are setting the table in early neurodevelopment in a way that has the possibility of leading to a wide range of outcomes. And I think it's now an incredibly difficult but an incredibly interesting question. Why does that happen? Why are some people more resilient? Why is the same gene that leads to severe autism and intellectual disability in one kid lead to mild language impairment in another? If we could capture that, we would be a long way, I think, towards some actionable hypotheses about therapeutics development. I didn't mention it, but we're starting to get a little bit of a signal with regard to Tourette and based just on gene ontology, but we're going to have the same problem. So our top gene is called CSLR3. It is a kind of classic planar cell polarity gene. The minute that you look at it, you go, oh, that's got to be a Tourette gene because it's involved in corticothalamic circuitry and dopaminergic neuronal development. You think that's got to be great, but the problem is that you can read 5 different papers, and depending upon when and where you mess with that gene, you can get completely different phenotypes. Interneuron phenotypes, you get a globus pallidus phenotype. If you turn it off later, you get an imbalance of excitability and inhibition. If you wait in development and then look at glutamatergic neurons. So I think for each of these things, it just stresses the notion that there is a fundamental difference between getting a hint about biology and actually then being able to move on to understand something more deeper. More deeper? Just deeper is enough. And this is really the idea behind, we're thinking about other ways to do spatial temporal analysis, again, leveraging really overlapping data sets, now looking at non-coding mutations that have the potential for a lot of spatial and temporal information. Enhancers, for instance, in the genome can be cell-specific and time-specific, so we're beginning to look for mutations that increase the risk in the same way that they do in the coding portion of the genome, but that will give us even more, we think of a Rosetta Stone in thinking about space and time in the development of ASD risk. And I think overall, it is a new world. When, as I said, for many years, I could tell you nothing. Every one of my talks was about how bloody hard it was to find genes for autism, and that we had not done anything in Tourette syndrome. And now it really is a new day in which we clearly know how to do this. And the next challenges are really to come together and think about how we can bring multiple different perspectives: systems neuroscience, computational neuroscience, basic molecular neuroscience, to leverage these, I think, very strong but still difficult clues to understand something that's going to impact our patients and their families. So thanks for your attention. I just want to make sure that, human genetics is absolutely a team sport. So the Ticks Genetics Consortium is co-led by a couple of great colleagues at Rutgers, Gary Hyman and Jay Tishfield, and involves a cast of thousands across the US and Europe. And then the autism work and the Tourette work couldn't have been done without Stefan Sanders, who I mentioned to you, and also Jeremy Wilsey, who as well was a student in my lab and now is a independent investigator at UCSF. So thank you very much for your attention.
So as you're doing more of these exomes and getting multiple mutations in the same gene, like have you gone back and looked at kids that have the same gene mutations and are phenotypes sort of emerging from those that are gene-based rather than just grouping them?
Yeah, it's a great question. So the question is, can we learn more about sort of the relationship between genotype and phenotype by going back after we've been able to use gene discovery? So the great thing about de novo mutations is that they carry very large effects, and the terrible things about de novo mutations, they're exceedingly rare. You get a lot of statistical power, but the most that we see any one of these genes is in less than 1% of kids that we see with ASD. So generating the power to do genotype, phenotype gets quite difficult. We've been able to do it for some of the copy number variations where the science is more mature. We just have many more people that have been looked at with that assay. And when we look there, we see very little coherence around any mutation, which actually, when you think about sort of how broad the outcomes can be from a single gene, that we can get autism or schizophrenia or intellectual disability or epilepsy, the notion that we might actually parse out subsets, syndromic autism, there'll be a handful of genes, I think, where that's true. For a lot of them, we're going to find that it's really opening up the door as opposed, I think, to narrowing it down.
Thank you for a great talk. In your Simplex cohort, have you ever gone back to look on the unaffected siblings as a proof of concept that they weren't affected by not the like some spectrum of ASD or other intellectual
Yeah, that's a great question. So we had the benefit of having a really exquisitely phenotyped cohort. It was, really the driving force in the Simon Simplex collection was thinking very hard about how to get something like a Simplex collection. There are a couple of answers. The first is they would not be allowed in without both categorical as well as continuous measures of social ability and autism. So they couldn't get into the study unless there was not evidence of ASD. If over time, because they are really closely linked to the Simons Foundation, as many families who participate in research in autism continue to come back again and again. If anyone gets a diagnosis of autism in a sibling, then they are flagged and they're moved out. So the vast majority of these families have a single child with a de novo mutation who's showing clinical evidence of ASD. Now, that said, we can infer genetic risk using different methods where we can look at common variation and say, is this a family that looks like they would have more risk from small effect common variants than others? And when we compare the Simons Foundation to control populations, we see that this entire set of families is enriched for common variant risk. We can't tell you the individual genes, but we can tell you this is not on a blank background. So what's happening is that there's a distribution of risk in the population that's probably something like normally distributed based on an accumulation of very small effect mutations. And what's happening is the de novo mutations are taking the kid in the family from wherever they would land, left to their own devices, and moving them out beyond whatever our kind of arbitrary threshold is for diagnosis. So they're not Mendelian risks. It's not Huntington's disease, where if you don't have the mutation, you don't have the disorder. It's a more complex genetic problem than that. And I think as we discover more about common variants, understanding the interplay between these rare large effect events and what we're all carrying around with us in terms of genes for social ability, et cetera, that's going to be another interesting set of questions we will be able to answer.
Okay, thank you very much.
You got one more? Or unless, yeah.
So I want your comments on antisense technologies, whether we could use it in a timely fashion and...
Great. So it was about antisense technology. I'm going to not answer that question, but make it more broad, which is, can you, once you identify the gene, would there potentially be technologies that you could intervene mid-fetal development? It's very problematic for a whole variety of reasons. So first is that, as I've showed you, this is not Huntington's. It's not a situation where you're certain of the outcome. Some of the kids with this mutation may either have nothing or may have, a little bit of language impairment. And are you going to try mid-fetal antisense, or some other therapy therapy in order to try to intervene very early on. That gives one pause. I think the flip side is, though, that what I usually get is people asking, well, this is like cause for therapeutic nihilism. If the problem is mid-fetal brain development, you're never going to get traction on that. And that, I think, is jumping, I think, to premature and I think likely erroneous conclusion. We can help kids with autism do better in their lives, and we can do it without knowing anything about the molecular and cellular underpinnings of what's happening by early intervention. If we do early intervention with speech and language, if we do early intervention with social interaction, we can see that kids improve. Their brains are plastic, writ large, not just at the synaptic level. And so we already know that, as I said, genes are not fate, and even if you have these mutations, your course is not set in stone. Beyond that, we know that many of these genes now also code for schizophrenia, which means that kids are going for 18, 20 years with carrying these mutations with no evidence. So something is leading to resilience. Something is allowing the system to buffer. And so I think that also suggests that the window is not necessarily closing in mid-fetal development. That's just where the risk begins to take shape. So I think that there are potentially multiple opportunities to think about reasonable interventions that would sort of capture whatever functional abnormalities there are. Even some element of this is clearly kind of quote-unquote strictly developmental. But I think that, I wouldn't be up here if I wasn't kind of a dewy-eyed optimist. So I do think that there are going to be opportunities to turn this into therapeutics.
All right,
thanks very much.